Righty. Okay, I think we're good to go. Hello, everyone. Good afternoon, and welcome to today's Tech Tuesday. Today, we're going to be talking about LabArchives, which is our electronic lab notebook for research. We're going to be joined by Michele Fortes today, Dr. Michele Fortes, sorry, who's our assistant professor of in the Department of Cell and Developmental Biology. He's also one of our senior liaisons for research in ITS. So Michele's got a lot of knowledge about both research and the many IT tools that we have to support you in your research. Before we begin, I just wanted to let everyone know we are recording today's session. So later on today, you should receive a follow-up email from me with not only a link to the recording, but also some other links to guides that could help you get started with LabArchives if you're interested in using it. And if you have any questions at any point during today's session, please feel free to type it in the chat. We'll be stopping periodically to take questions, but you can ask it at any time there. And if you have maybe a more complicated question, you can hit the raise hand button at the bottom of your screen. So if you want to come off the mic to kind of, provide more context, that's fine too. So without further ado, I'm going to hand it off to Michele. Thanks, Michele, for joining us.
Yeah. Do you want to wait one more minute? Because there is a lot of people still joining, I think.
Sure. I mean, or you can, if you want to give like a quick intro, you could do that too while people are joining as well.
Okay. Hi, so my name is Michele Fortes. As Vanessa said, I'm an assistant professor. I'm in the Department of Cell and Developmental Biology, and I'm also the senior liaison for research in ITS. And so here's my email. Feel free to email me and for any questions or anything. And I've been giving a lot of these presentation also to individual labs. So if you want to organize a presentation of LabArchive for your lab, just let me know, and I'll be glad to set up to come to your lab meeting and set it up and make sure everybody is comfortable with using it. Okay, so this is the most important slide, just everything condensed here. So if you remember only one slide, maybe this is the right one to remember. So first of all, it's a free account for all WCM faculty, staff, postdoctoral students. So everybody has an account as soon as they join. And the place to log in here is mynotebook.labarchives.com. I will show you in a little bit. We're going to do actually demonstration of things. Okay, so LabArchive, very importantly, we choose a system that works on Macs, on PC, on Linux. It's completely cloud-based. And very useful, also works on smartphones and on iPads, smartphones, both iPhone and Android, is to type entry. Maybe it's a little bit difficult on the smartphone, but what the smartphone has, which is really useful, is you can connect the LabArchive software on your smartphone to the camera of your smartphones, and then you can take pictures of gels, take picture of your lab notes, take picture of graph, take picture of images, X-ray, everything. And then all the work is stored in the cloud, is searchable, is backed up, and is accessible from everywhere. So you don't have to be in your lab. You can work from home. You can enter things into your lab and then go look at them when you are at home, make changes and everything. And the searchability is very important, because basically every word, everything that you type in LabArchive, you can search for it. So if you want to know where you used an antibodies against the PIC2, or when is the last time you did the mRNA looking for CSARC, you can find it immediately. And everything is backed up. So you don't have to worry about keeping multiple copies of what you're doing, as if you use some other electronic storages. The total storage per user is unlimited. At the moment, the individual file size maximum is 4 gigabytes, but we're going to increase it pretty soon to 16 gigabytes. And since you can have more than one notebook, so each individual can have many notebooks, what LabArchive suggests is not to have more than 40,000 entries per lab, and try to keep it below 1 terabyte content per lab, just to facilitate the search. Two more important things is one, that in the lab, each individual researcher should assign the ownership of the notebook to the PI. I'm going to show how to do it. And the other important thing is, yes, you can store PHI in LabArchive. Now, the only caveat with that is that since LabArchive makes it very easy to share your own notebook or a portion of the notebook or a page of your notebook with somebody else at Cornell or outside Cornell, then of course you have to make sure that what you're sharing, if you share something that contains PHI, those people have to be authorized, be on your IRB protocol or other things to make sure that you're sharing with somebody who has permission to access those PHI. Okay, why do you want to use LabArchive? There are three major reasons, I would say. The first and most important, in particularly recently, is that it helps you with your data retention mandates, both the NIH policy and WCMC policy. So if you store your data in LabArchives, you are complying with both the NIH and the WCMC policies, and you just have to tell the NIH or Weill Cornell that you're keeping the data in your LabArchive notebooks. The second thing is there is a lot of talk now of the reproducibility crisis, and in particular, another problem is lab members leaving, and that's why I suggested that it's very important that the lab members, transfer the ownership to the PI, because what often happens is a lab member leaves, the notebooks are very difficult to read. It's difficult to go through all the pages to find whatever the next person that wants to reproduce an experiment wants to do. And so if you have everything in LabArchive, everything that one of the members has done stays with the PI. The PI can actually give access to the person who is leaving if they need to finish a paper, need to finish a manuscript, and they can look at the lab and the LabArchives notebook for free from any email, either from the institution that they're going to, sorry, but also from a regular Gmail access. And then the other thing, which is also useful, since everything in LabArchive is time-stamped and recorded, if you make changes, LabArchive records the changes. So it's very useful for a patents application, also to defend yourself allegation of misconduct. So again, this is very important. Transfer ownership to the PI. So the data is retained, the PI is accessed, and then you can continue to access it on your own. Oops, sorry. Okay. What's going on? As I said, each notebook entries have an indelible timestamp, and these are going to show you how to use it. And again, everything is stored in the cloud, is searchable, and is backed up. And I just told that the PHI, you can use it for PHI. So how do you want to organize your notebook? There are different alternatives depending on your preference. I have a couple more examples actually. This is an example from Indiana University. So most people have a user, and they have most people organize it by project. So you have a shared lab notebook with protocols, templates, notes from lab meeting, etc. And then one notebook for each of the projects. and since you can share the lab notebook with everything, it's very easy to create that shared notebook. So one of the each notebook can have different roles. So the most important role is the notebook owner, and then I said it should be the PI. And then there are the users who access the lab. Basically the researchers in the lab who's using the lab notebook. You can have an administrator, possibly the lab manager, and then you can invite other people as guests. So if you collaborate with somebody else at Cornell or even outside of Cornell, you can give them access to your notebook. You can define what preferences, what privilege that this person has. So say you're doing a set of experiments with someone else, then you can have you create a notebook, you put all the protocols of the reagent, and then you share the pages with these people, and then you give them access so they can write their own experiments in the notebook. So you have this possibility of having guests and multiple users is very, very important. So this is another example of for a larger lab. So again, the notebook owner is the API, then people have individual notebooks, and then somebody, usually the lab manager, creates a resource notebook, and then it shares it with every user. So say in this notebook here shared, you put the lab protocols, you can put where all the reagent is, you can put the material safety data sheets, you can put how to make the reagents, the formula for reagents, where all the reagents are in the lab, in the freezer and everything. And then you can have also another, if 2 or 3 people collaborate on a project, then you can have another notebook specifically for the project and share it with other people. So the structure is very flexible because it's very easy to invite, it's very easy to create multiple notebooks per person, and it's very easy to create, to add people as users. So this is up to your lab. And again, I said, if you think it's useful, I can come to your lab meeting and we can discuss this a little bit in advance. The other very important thing on LabArchive is you can integrate it with a lot of, sorry, for external software. So you can have some experiments you can directly come from equipment. These are a little bit limited. Of course, you can do direct entry, you can do email attachments, so you can email it to your lab notebook and it's going to get stored. You can take things from the digital camera, pictures, from mobile devices. You can use you can create forms and templates. So instead of rewriting every day the same step for the experiments, you create a template and say, okay, step number 1, dissolve the cells in PBS, things like that. So you can create a template for your experiment, and you just make the little changes that are specific for your experiments. And then you can also get things from the LabArchive. Sorry, you can get also things from OneDrive. It connects directly with Microsoft Office and Prism. So you can download a plugin, and from Microsoft Office, you can directly put things save it directly into your lab notebook. Same for Prism. I'm going to show a little example. Connects with Jupyter very well, connects with Box and OneDrive, of course. So there is a very large variety of data files and data format that LabArchives understand. And then you can output it, of course, either in PDF or in HTML. There is an option for accessing LabArchives from our desk. So I'm reading a question. Maybe Johannes, you want to unmute yourself? Johannes and so no, you have to use it from the desktop. You have to log into the the browser every time. The only caveat to that is there is an application that you can download it to and I'll show it at the end. There is an application that you can download, which is called Folder Monitors. You install it on your Mac, on your PC, and what it does is looks into certain folders that you determine, and as soon as it sees that there is a file that is changed in that folder, it uploads it in LabArchive. So you can have a folder, say, called gels, and then whenever you have any new gels, you upload it you put it in that folder and then gets uploaded automatically. But there is no desktop offline application, if that's what you're asking. Did I answer the question? We don't know.
Michele, there was also another question I got sent to me, whether LabArchives is suitable for bench research or clinical research.
It can be both. So I mean, it's basically just a lab notebook. So it's a notebook. You will see with the interface, they did it maybe even to an excess just to replicate a notebook. But you can see that it can integrate with RedCap. So, but it's just a notebook. You can configure it as you want. You can add all the templates that you want. There are a lot of things called widgets that you can add. So you can use it both for clinical research and bench research. Okay. So let's give a little bit of a demo. So you go to my notebook, labarchives.com. On a Mac, I'm using a Mac. On a Mac, I think Chrome is you can use it with Safari, but Chrome is a little bit better because it's easier to do control clicks. So I go my notebook, labarchives.com. Okay, if you are inside Cornell, you see that is already chosen while Cornell. If you are outside, it's going to show this very, very long list of all the places which are already using LabArchives, and there is Einstein, Caltech, Carnegie Mellon, Cedars-Sinai, Columbia, Cornell, Duke, MIT, Brigham. I mean, the number of the places that are using LabArchives gets bigger and bigger by the week, especially recently, because now the fact that if you use LabArchive, then you comply with the NIH retention policy. That's is a very big plus. Okay, so you go to an institution, you enter your CVID and password, and there you go. So this is the one of my notebooks, and it goes by default. It goes to the most recent notebook you've used, and here there is, you can see there is a little 6, and it says that I have 6 different notebooks. If I go here, I can look at my other notebook. So, and again, I can have an enormous number of. You can have as many notebooks as you want. Okay, so I'm going to try a new notebook, and here you have. You see that the way I created this notebook, you can create folders. You just simply click here to add a new folder, and you can do like I used to do it with paper notebooks. So there is one folder for protocols, one folder for standard operation procedure, MSDS, templates. Okay, let me show you what I did. So here I have create one folder with each agent, and you can see here, I don't know if you can see it, but there are the 2 little human figurines here. This means that this folder is shared with somebody. So I created a folder with all the reagents. where my things are. And so I have a page with all the freezer boxes. And so these are all the freezer boxes. These are a certain number of freezer boxes. And if I want to create add the new freezer boxes here, I used it recently. There are these widgets. And these widgets are all little like mini applications. So you can do bacterial grow calculator, buffer calculator, which is very useful. Chemicals, everything. And I choose a freezer box. So I can add the new freezer box. I can choose the size of my freezer box. And then in each individual well, you can enter and say what's there, and you get to keep track of things. And so once I order this, so now if I'm looking for a reagent, so say I'm looking for the PIC2 antibody, I just search my notebook, and it tells me everywhere that I have PIC2 antibody. So this is very useful for keeping stock of where all the reagents are. And again, these are shared with other people. And I'll show you in a sec. The way you, sorry, the way you share it with other people, you right click, you go share, and so I can invite somebody, and I can invite to view or to edit. So say, in this case, I'm going to invite to edit. So because it's taking a little bit of time to check for all the people. I just show you. So there, I would add a new person here. And here I can see the people that already give access. So Vanessa already has access. So now if she wants, she can go to notebook, and then this folder is going to show up in her lab notebook. Okay, and so protocols, reagent, templates. Templates are very useful because one thing that I used to do all the time is isolate neutrophils from blood. And so here I have a blood, I have a Word document with all, and notice that that's a Word document, and I opened it directly in LabArchive. You don't need to open it into Word. Word, LabArchive has a Office editor inside. And so here are the steps that I normally use to do this. So I'm gonna close it now and go back to my, so I just to show the template. And then say I want to do a new experiment, I'm gonna add a new page with today's date. I mean, there you can put experiment number, you can put date. Everybody has a different way to record. But so I add a new page with today's date, and then I'm gonna put a template in. Oops, sorry, no. Sorry. Here's my template. Okay, sorry, something, I apologize. But I mean, you can create a template and then use the template and put it here. Okay, no, I lost my name. Sorry. Okay, and then you see that in a page. So you can enter text. we can have, here it is the ARC document, and then I can put, I can input gel, I can input spreadsheets, I can add this one of the widget for the dilution calculator and everything. Now notice that every entry that I have here is timestamped. And then look at this. So I can, this is a picture, but I can edit the picture. And so I can add labels to the picture. If I want, let me make it a little bigger. Okay, this is good. I'm gonna save it. Very slow today, not sure why. Sorry, but anyway, here it is. So now it's labeled. If I click on these three dots and I check view revisions, I can see all the things that I've done to this video. So this is, again, is very important for good data retention policy, because you can always go back to a previous version. So I say the first time that I did something was on August 2021. This is my name, and where it says where I did it from, to which the IP of the computer where I did it from. And then I made an annotation, then I reverted, I deleted, I made another annotation. If you want, you can always go back to the original version. So any change that you make is kept track of. And this is very important, in particular if somebody makes changes and you don't recognize the figure, say who made these changes, it tells you who made the changes. It's very important for IP, it's very important for if somebody questions your data. Okay, so again, the other thing I want to show is, again, I said that is very important, is to transfer ownership. So what you do is you click here on these three dots and you go to notebook settings. Okay, and then I go to user management. Okay, so now it tells me who is the owner at the moment. Who are the other people that have access to my notebook. And so Anna has guest access, so she can read my notebook, but she cannot make changes. Vanessa has user access, and she has full access and can make changes. If you want to make any changes, you can delete people. Sign and witness are, there are particular specialized features for which are important for patent application. So when you finish the page, you can sign the page. The page cannot be changed anymore, and you can have somebody else witness your page. So in this case, I want to change Vanessa. I'm going to transfer ownership to Vanessa. So I click here, and I'm going to select Vanessa. I'm going to transfer ownership to Vanessa. Assuming that Vanessa is the PI, I'm going to say, okay. I'm not going to do it now because I still need to show things, but, and so actually I'm going to do it, okay. Okay, so at this point, you can see that Vanessa is the owner. And so even if I leave, then Vanessa still has access to the notebook and to everything I've done. And then I'm still the administrator because I created, so I can basically do everything. But Vanessa is still the owner. I can invite other people, I can share it with everybody. and I can add notes. I can do everything, but Vanessa stays at the owner. So you can see that here, my view has not changed very much. Any page that I was before, is still I still have the same access. So so create a new, how do you create it? So if you want to do something new, what do you create? So you create a new page. I just show you. And then you can, what you can do, you can just actually start typing if you want. So you add what? You can add rich text, you can add plain text, and just start typing. When it stays there, I can attach. I can use rich text if I want. These are the. I can attach an Office document. I can attach a widget that again, as I show you, there is a lot of different widgets for RNA calculator, buffer calculation. I can have the Google Docs in there. I can add the Google Calendar. The amount of things that you can attach is basically limitless. And every time I do something new, again, there is a timestamps, and I can see the whole history of the whole thing. So it basically it replicates exactly what a paper note. Now, if I again remember that there is you can use things also from your phone, so you can take a picture of a gel, and it gets. Let me do a little I'm gonna show you. I cannot show you, but you can see the. Okay, so I open. I don't know if you can see, but I open LabArchives on my phone. This is the macrophages. It's a new page. Okay, so I see here. I want to take an image. I want to take a photo. I'm just gonna take a picture of something. Let's see. Yeah, I have some pictures already there. Okay, it's uploading the thing, and then if I reload, here is the picture I just took, just a FedEx image. So it's very, very easy to take picture of gels or other things. Okay, Steven wants to know, to meet NIH sharing mandate, is possible to have a selected number of notebooks for sharing with the world? Yes, it's not ready yet at the moment, but yes, you could do that. Yeah. So if we are setting it up now. So the simplest thing is that's a little bit tricky because you have, I think the best solution would be to create a new notebook with the things you want to share and just share those rather than sharing your main notebook, because there is going to be a tons of pages that you don't want to share with the whole of the world, like templates, ideas, lab meeting notes, things like that. But yes, you can select a notebook and share the notebook. Yeah, we are ITS is working on it. It should be available pretty soon. Okay, I think I showed the most important things that I wanted to show. Okay, so a few tips. Yeah, you can import everything. Import spreadsheet that contains chemical database. So if you currently you have in your lab you have Excel spreadsheet with all the reagents that you're using, you can in an Excel spreadsheet, then you can import it in one of the widget, and it's going to show into a nice table, searchable. And this is the link, and you can go there, and it's going to show you all the instructions. Okay. The sketch and annotation tool, so is this tool here. So now in these labels, I can sketch and I can add whatever I want. You can make labels and everything. And again, you can create or edit Microsoft Office document within your notebook directly, and also integrates fully with Google Docs and Google Sheets. You can also download the MS Office plugin if you want. And what it does is, let me show you, I just launched Note here. Okay, so this is a new Word document. And here, once you install the plugin, here, you can open and save things directly from LabArchives. So now this document, I can go and save it directly. Let's see, in today's page. And so then if I go back to here, and I refresh it, it's going to show the Word document I just created. I can just look at it and use it as I want. Same thing with the Prism. If you have a graph in Prism, you can go to the File menu, and it's connected to LabArchives. So I can open any file from LabArchives. I can. These are my files that I put in LabArchive, and these are my notebooks. So this one is one that I saved in LabArchive, and I can reopen it and make any changes I want, and then store it back in LabArchives. To connect one thing that when you install those plugins or when you try to connect Prism, it's going to ask you for your email and password. Note that the email is the same at Cornell. The password is not actually your Cornell password, but you have to get there is a specific password for LabArchive, which is a very long string, which is here, external app authentication. And there is thankfully, you can just copy this. And then you paste it in Word or Prism or anything else. Okay, and then the last thing I wanted to talk about is this LabArchive folder monitor. And this is very useful because you can upload files automatically. So you can here you can download the folder monitor and let me see if I have a folder monitor. Okay. This is what you download from LabArchive, and I'm going to install it. Okay. I have my folder monitor. Folder. It's called LabArchive. Yeah, LabArchives folder monitor. So now this is my email, but the password is the password that I copied from LabArchives. And so I click OK. I want to connect. Okay, and so I don't know if you can see here, but basically what you do is you make rule edit rules, and you can you select a local folder. So say everything that I put in this in the data folder. Every time there is a new file, and I can choose all the files or just image files or everything, and then I tell them where do I want it. Where do I want to put it? So I want to put in this in the test notebooks for project 1. He asked me if I want to enter every day I create a new page, yes or no, how many max to enter per page. And so now every time I put something in the data folder, automatically puts it in LabArchives. Okay. Vanessa, do you have any more questions?
Sorry, I was trying to unmute. I don't see any more in the chat, but if anybody has any questions, we have a couple minutes left. Feel free to put it in the chat, or if you want to raise your hand so we can unmute you and you can ask a question over the mic, feel free. Oh, we just got one from Shahida. Where will this recording be posted? Oh, Shahida, so I'm going to be posting this on our YouTube channel, but I will be emailing it to you. So whatever email you use to register for this session, that's where I'll be sending a link to it. So you'll definitely receive a copy of the recording.
Okay, and if you want more information, again, so you go to the ITS website, you just search for LabArchives, and you find the page which has a lot of different resources. I can show you right now, actually. Okay, click on use LabArchives. There are LabArchive have their own YouTube page. They're very nice short movies. The database, the quick start guide. You can log in for seminar. You can LabArchive support. You can download things. So there is a lot of way to get information. Again, please feel free to email me if you want. This is my email, and I'll be very happy to.
Mikhail, we just got a question. If you're only using the Excel/RedCap for clinical research, would this offer any advantages or disadvantage to switching?
Well, the advantage to switching, if you only use Excel and RedCap, so those would not comply with the NIH policy. So you have to have it with LabArchive, so the Cornell policy states that every time you have to keep your data somewhere, and so LabArchive complies with the Cornell policy, and it complies also with the NIH policy. If you just keep RedCap, you can connect it to your LabArchives. If you use Excel on your hard drive, that's not a good place to keep data for many reasons. First of all, because it's transient, it's not a permanent place. And second, the Cornell policy states that when every time you have a publication, every time a grant finishes, you have to archive the data in something called WIDR, WIDDR. And you have to upload it there if you want to use Excel. If you use LabArchive, you can just link it to LabArchive, the entry. So in particular, Excel, I'm quite worried about using that. RedCap is in the cloud, so I think it's a little bit safer. By using Excel, saving stuff on Excel on your computer only, I don't think it's really a good idea, a safe idea.
Thanks, Michele. And we just got a note from Tom Campion. I can clarify on RedCap.
Yeah, thank you. Yeah. Tom, you can unmute if you want.
I'm muting him right now. Tom, if you want to unmute, feel free to clarify on the RedCap comments.
Terrific. Michele, thanks for doing this overview today of LabArchives. I learned a great deal, and very happy to see that so many people attended and learned a great deal as well. As far as RedCap goes, and thinking ahead to things like the NIH data management and sharing policy, please continue to use RedCap. As Michele pointed out, do not use Excel. Excel is incredibly powerful, but we find it to be best for data analysis rather than data storage. With the data collected for your project in either RedCap or LabArchives, it provides a great segue then to being able to upload your data somewhere else. So one of the benefits of RedCap is we're continually saving the data, backing up the data, enabling disaster recovery in the same way as LabArchives. And at the end of a research project, we can then work together to maybe remove any other sensitive elements that we might need to before we upload the data, say, to a public repository. Or we could take that very sensitive data set that may exist in RedCap, export it, and make it available in the wider resource that Michele was describing, and that's only for internal purposes. So things that we can figure out, but please continue using RedCap and LabArchives. Does that make sense?
Yeah, absolutely. I agree 100%. I mean, you can use Excel as long as you store the files in LabArchives. Right. So Excel as a computing tool is perfectly fine. The problem is where you store the data. So as Tom said, keep the data in RedCap. If you want to do any elaboration or and remember that LabArchive is good for PHI, and then you do whatever you want to do in your Excel spreadsheet, and then upload it in LabArchive. So you have all your data is either in RedCap or in LabArchive, and it's perfectly safe.
Thank you, Tom and Michele, for clarifying that. I don't see any other questions, so I'm going to wrap up this session. However, if you find that you do have questions later on, whether it's about LabArchives or NIH data storage policies, please feel free when you get the email with all the follow-up information. McKaylee's already offered his email. I'm going to put that into the follow-up message. But feel free to just write back if you have any questions, and we'll be happy to help you get started with LabArchives or any other questions you have about storing your data. Thank you guys so much. Thank you, McKaylee.
Thanks. Bye, everybody. Thank you for joining.